All right, let's just dive right in. Picture this. The inside of a single one of your cells is like this non-stop, microscopic city. It's a whirlwind of activity. Materials moving, structures being built, waste getting tossed out. But, just like any city, all that movement, all that transport, it costs energy. And our cells have come up with some seriously clever ways to manage that energy budget.
And that really brings us to the big puzzle we're tackling today. I mean, think about trying to push a huge boulder up a hill. That takes a ton of direct effort, right? Well, cells face that exact same kind of problem all the time, trying to move molecules from a place where there's not many to a place where they're already crowded. They're literally pushing them uphill. So the question is, how on earth do they pull that off without paying the energy toll for every single push?
So here's our game plan. We're going to start with the energy puzzle itself. Then, we'll get into the secret of how molecules can hitch a ride, we'll look at the different ways they do it, same direction versus opposite, and then we'll check out a related transporter that prefers to go solo. And finally, we'll tie it all together and see why this tiny little cellular hack is just so critical for your health.
Okay, Section 1. Let's talk about the big challenge. For any cell, one of the toughest jobs is moving stuff against what's called a concentration gradient. Honestly, you can just think of it like trying to swim upstream against a really strong current. In nature, things just want to spread out, right? Move from crowded places to empty places, high concentration to low. But to do really important jobs like absorbing the nutrients from your food, a cell has to do the exact opposite. It has to cram molecules into a space where they're already packed in tight. And that process, well, it takes energy. And that's where things start to get really clever. So, how does the cell crack this problem? Well, it uses this incredible trick, almost like a biological carpool system. This is what we call the no direct ATP solution.
And here it is, the official name, secondary active transport.
So instead of using energy on the spot, the cell is cashing in on energy that it's already stored up, kind of like a rechargeable battery. It spent time building up this powerful gradient of ions, and now it's going to use that stored up potential to get some work done.
And this is the most important part to remember. Unlike other pumps in the cell that have to burn the main energy molecule ATP for every single movement, this method is indirect. It's like getting a 2-for-1 deal. It's a brilliant piece of energy recycling that makes the whole system unbelievably efficient.
Now this slide is fantastic because it shows you exactly how it works. It's a two-step process. First, the cell makes an investment. It uses those ATP-powered pumps, what we call primary active transport, to shove a whole bunch of ions, usually sodium, out of the cell. Think of it like pumping water up a hill and storing it in a reservoir. You're building potential energy. Then, step 2, it opens a gate. As those sodium ions come rushing back in, flowing down the hill they were just pumped up, that powerful flow of energy is harnessed to drag another molecule, say glucose, right along with it. It's an amazing piggyback system.
So, this hitching a ride idea is all thanks to these specialized proteins we call cotransporters. And it turns out they come in 2 main flavors. And honestly, it's beautifully simple. You've got your symporter, where both molecules travel in the exact same direction. Just imagine them linking arms and strolling into the cell together. Then you have the antiporter, which is more like a trade-off. It moves one molecule in, while at the exact same time, it pushes a different molecule out. Let's look a little closer at the symporters. This protein is sitting there in the cell membrane, and it's designed to grab 2 different molecules at once. The energy that gets released by the one molecule flowing downhill with its gradient, well, that's the power source that shoves the other molecule uphill. The revolving door analogy is perfect. One person does the pushing, but a second person gets to sneak through at the very same time. And hey, this isn't just some abstract concept. The sodium glucose symporter is a real-life superstar in your body. It's literally how the cells lining your intestines snatch up all the glucose from the food you eat. Sodium rushes in, and it drags that precious sugar in right behind it. In fact, understanding this one mechanism has led to huge medical breakthroughs, like the SGLT2 inhibitor drugs that help treat type 2 diabetes by targeting these exact transporters. Okay, now for the other type, the antiporter. Like we said, it's an exchange. A perfect example is the sodium-hydrogen antiporter. It allows a sodium ion to flow into the cell, and in return, it boots a hydrogen ion out. This is absolutely essential for keeping the cell's internal pH balanced and stopping it from getting too acidic. So, we've seen how cells can cleverly move 2 molecules at once. But what if they only need to move 1? Well, that's where our next protein comes into play, and it's the big exception to this whole cotransport rule. This is the uniporter, and it's as simple as it sounds. One protein is responsible for one type of molecule moving in one direction. That's it. No coupling, no carpooling, no tag teaming. So a uniporter is basically just a facilitator. It's there to help a single kind of molecule get across the membrane. Usually this is what we call facilitated diffusion, and the key thing to know is that it helps the molecule move down its gradient, from high concentration to low, not against it. It just opens the door and makes the path easier. So while it's a super important transport protein, it's not a cotransporter because it's not using one molecule's energy to move another. It's the solo artist of the cellular world. All right, we've gotten through all the mechanics, but now let's zoom out and connect all of this back to the big picture. Why is this tiny, microscopic energy hack so incredibly important for every single one of us?
This quote right here just says it all, doesn't it? It gets right to the heart of the matter. That powerful sodium gradient that your body is constantly working to maintain, that's the battery pack that powers the transport of essential stuff like glucose right into your cells.
Exactly. I mean, think about it. Every single time you eat breakfast, lunch, or dinner, there are millions of these tiny molecular machines whirring away, using this secondary active transport system to make sure you get the energy and nutrients you need from your food. It is absolutely fundamental to how we live. So let's just do a quick recap of the most important points. Secondary active transport is this amazing energy-saving trick that runs on the stored power of ion gradients. It's super efficient because it doesn't burn ATP on the spot. You've got symporters and antiporters doing the work, moving molecules either together or in a swap. And at the end of the day, this whole cellular hack is essential for life. It really makes you wonder, right? I mean, our cells are just packed with these elegant, incredibly efficient solutions that have been perfected over billions of years of evolution. If they figured out a system this clever just to move some molecules around, what other amazing tiny engines are humming away inside of us right now, just waiting for us to discover them?